Right now we have the most important neat diagrams in NCERT class 11th biology. So first we can see the stomata of the bean shaped guard cells, A diagram, and outermost is the epidermal cell. Next we have the subsidiary cell, then chloroplast which is green color round shaped. Then we have the bean shaped cell, the guard cell, at the inner stomatal pore is shown. So revising once again, outermost is the epidermal cell. Then we have surrounding the guard cell is the subsidiary cell. Then we have guard cell which is bean shaped. Then chloroplast in it. Then stomatal pore. Next, figure B, stomata with dumbbell shaped guard cell. So we can see the outermost is the epidermal cell. Then we have the dumbbell shaped cells which are guard cells. Surrounding it we have subsidiary cells. So this was all about dumbbell shaped cells in monocot. Next diagram is of dicot leaf. So we can see the thick layer of bundle sheath cells surrounding the vascular bundles. We have xylem which are shown by rounds. Then we have the phloem distributed in between it. Next we have adaxial epidermis, and below we have abaxial epidermis. Then the space is called stroma. We can see the substomal cavities. Then we have the spongy mesophyll. Then we have air cavity, and then we have the palisade mesophills. Next is the monocot leaf. So upper layer is the adaxial epidermis. Below we have the abaxial epidermis. The space is called stoma. We can see the phloem and then xylem. The mesophyll cells are rounded cells with thin cell wall. Next we have the dicot root. So we can see the root hairs, then epidermis, then the parenchyma cells with some space in it. It is cortex, then endodermis, then pericycle, then protoxylem, and centrally faced metaxylem. Then between it we have pith, and then phloem. Next is the monocot root. So we have the root hairs, epidermis, cortex, endodermis, and then pericycle. So in this both the cases, pericycle comes after endodermis. Then we can see the phloem, then protoxylem, then metaxylem, and then pith. So here in the diagram of vascular bundles, we can see the first one is the radial, next one is the conjoint closed, next is conjoint open. In conjoint open we can see the cambium present. First diagram can be seen in roots, second and third in stem and leaves. Next we have the transverse section of monocot and dicot stem. So starting with dicot stem, we have the epidermal hairs, then epidermis, and then hypodermis, then parenchyma, then endodermis, and then pericycle, vascular bundles, and in between the vascular bundles we have medullary rays, and the center has the pith. Next in the next diagram of dicot stem, we can see the outer is the epidermis. Then we have collenchyma, then parenchyma, then endodermis, then pericycle, phloem, and then cambium, metaxylem. Centrally we have protoxylem, then pith. Next diagram of monocot stem, we have outermost epidermis, then hypodermis, Then vascular bundles, centrally placed vascular bundles are larger as compared to the peripheral. Then ground tissue can be seen. Next diagram of monocot stem, outermost is the epidermis, then we have hypodermis, then vascular bundles are shown, then phloem, xylem, then ground tissue can be seen. Next diagram, meiosis 1 in cell cycle and cell division. So first we can see the prophase 1, then metaphase 1, anaphase 1, and then telophase 1. After telophase, two cells are formed in meiosis 1. Then we have prophase 2, metaphase 2, anaphase 2. Note the difference between anaphase 1 and 2. Then we have telophase 2. The stages after meiosis 2. After meiosis 2, 4 cells are formed. Next we have early prophase, then late prophase, then we have transition to metaphase, then metaphase. So these are the stages of mitosis. Rest we have anaphase, then we have telophase, and this is very important, this is interphase. So revising once again, this is interphase, this is telophase, this is anaphase, then metaphase, chromosomes lie at the center, this is metaphase, then transition to metaphase, in late prophase, early prophase. Next is a neat biology animal cell diagram. So we can see here cytoplasm, then centrally we have nucleus, then the dark blue color shaped is the nucleolus, and surrounding the nucleus is the nuclear envelope. Then we have the Y-shaped structure which can be seen in the diagram, which is smooth endoplasmic reticulum. Okay, next you can see the rough endoplasmic reticulum in which ribosomes can be seen. Then we have the mitochondria diagram. Then the round shaped structures are ribosomes for protein synthesis. Then the small round is the lysosome. Then peroxisomes also can be seen. And in animal cells, centrioles are seen. Then we can see the plasma membrane. Golgi apparatus is also seen. Then microvilli. Next, we have plant cell. So we can see the chloroplast is seen. The centrally placed big vacuole is seen. Then we have middle lamella. Then cell wall. And then chloroplast can be seen. Yes. The cytoplasm we have the round shaped peroxisomes, microtubules. Then plasmodesmata also transverse the cell wall. Next, smooth endoplasmic reticulum, then rough endoplasmic reticulum with ribosomes, then nucleus, the centrally dark blue colored is the nucleolus, Golgi apparatus, which are same in both, then nuclear envelope, then plasma membrane. Yes. Next we have endoplasmic reticulum. So in endoplasmic reticulum we can see the nucleus is present, then we have nuclear pores are present, then you can see the small round shaped ribosomes are present, and the structure without ribosomes are smooth endoplasmic reticulum. Then structure with ribosomes are rough endoplasmic reticulum. RER is responsible for protein synthesis and SCR for lipid synthesis and secretion. Next, this is important. So it is Golgi apparatus. Cisternae is seen in Golgi apparatus and cristae is seen in endoplasmic reticulum. Next diagram is of endoplasmic reticulum. We can see the outer membrane, then inner membrane, then matrix, fluid filled matrix, then inner membrane space. Then we have crista, folding are called crista.